Hello there, welcome to my poster. My name is Yingxue Ren. I'm an assistant professor at Mayo Clinic. The title of my project is Alzheimer's Risk Factors, Age, APOE Genotype, and Sex Drive Distinct Molecular Pathways. Now let me first show you our experimental design. We had human APOE2, E3, and E4 target replacement mice, both male and female, at 3 different ages, including 3 months, 12 months, and 24 months. From these mice, we harvested brain samples and performed transcriptomics, and we harvested serum samples and performed metabolomics. From these data sets, we then did WDCNA, or weighted gene co-expression network analysis, and this allows us to identify gene or metabolite modules. We also performed differential expression analysis, which allow us to identify genes and metabolites that are differentially expressed between conditions. And from this, we identified pathways that were enriched by these genes or metabolites. The first thing we did in our analysis was to look at global characterization of brain transcriptome and serum metabolome profiles in our mice. So on the top side of this figure, panel A and B shows the brain transcriptome data. So here we can see that age had the strongest impact on the gene expression profiles. This finding was recaptured in panel B by the TSNE plots of our samples. On the bottom side of this figure, panel C and D shows the results of serum metabolome, and here you can see that APOE had the strongest impact on the metabolome profiles. And this finding was recaptured in panel D by the TSNE plot. This figure here shows the impact of APOE genotype, age, and sex on gene co-expression networks of the mouse brain transcriptomes. Panel A here shows the module trait correlation between modules and our traits of interest. So each row here is a module, which are groups of genes that are highly correlated with each other. And the 3 columns represent APOE, age, and sex. For example, you see that module light cyan is highly regulated in APOE4. And among the top 10 module hub genes of light cyan module, you'll see that many of them belong to the SAPINA3 gene families. And this module is enriched in pathways such as peptidase inhibitor activity. Now module blue and module pink are elevated in aging samples. And here you can see the top 10 module hub genes in module blue and module pink. And correspondingly, you'll see that module blue genes are enriched in extracellular vesicle processes, and the module pink genes are enriched in immune system process. The next thing we did was to see whether the mouse modules were preserved in human. For this, we looked into 3 public human data sets, including the ROS-map human prefrontal cortex, the Mayo Clinic human temporal cortex, and the Mayo Clinic human cerebellum. The figure panels A, D, and G here shows the module preservation, and the dotted line of 2 represent preservation of modules. So here we can see that module blue and module pink are preserved in human in all 3 data sets, but module light cyan was not. And we think that this is because module light cyan was mainly driven by SAPINA gene families, and in human there's only 1 SAPINA3 genes. To confirm this, we looked at the SAPINA human gene expression in E4 carriers versus non-carriers. We also looked at another module hub gene, ATF4. In temporal cortex, we saw a significant difference in expression between carriers and non-carriers, but this difference was not significant in cerebellum. We next identified differentially expressed metabolites, or DEMs, in the mouse serum. Panel A shows the differences between APOE genotypes, and you can see that E2 is significantly different from E3 and E4. And on panel B and C, we show the top 2 DEMs in terms of APOE genotype. Panel D and E and F shows the differences between age, and you can see that 3 months is different from 12 and 24 months. And panel G, H, and I show the difference between male and female, but here the difference is less pronounced. Similar to what we did with the brain transcriptomics, we also performed WT-CNA analysis on the metabolome. We identified modules that are correlated with APOE, age, and sex. And if we look at, for example, module yellow, this module is upregulated in APOE2. And here on panel D, we see the top 10 module hub metabolites, which are a group of PCs. Now finally, to highlight our findings, among Alzheimer's related risk factors, we found that aging leads to the most profound changes in brain gene expression network, highlighted by the immune module with a significant upregulation of AD risk genes TREM2 and TYROBP. Extracellular vesicle gene network is dysregulated with aging in the brain. Alzheimer's risk allele APOE4 broadly increased expression of SEPINA3 gene family. And finally, Alzheimer's protective allele APOE2 drive unique serum metabolome profiles.